Welcome to Hope, Innovation, Discovery, a podcast produced by CURE, Citizens United for Research in Epilepsy. In today's episode, we'll hear from Dr. Julie Milder, Associate Research Director at CURE Epilepsy. Dr. Milder recently visited CURE grantees in the Pacific Northwest. She shares encouraging news about 2 research studies showing tremendous progress in brain injury seizure control. Julie, you recently concluded some site visits with several CURE-funded researchers out in the Pacific Northwest, and I wondered if you could tell us a little bit about your trip and perhaps lead off with the work being done by Dr. Detlev Boysen out at Legacy Research Institute.
Sure. So as a part of our strategic plan for the year, we were each challenged on the research team to visit at least 2 grantees in their labs. We've never done this before as a part of our standard practice, but the idea is that by going to visit them where they are with their people, we could get more than we would learn from a written progress report every 6 months. And so I first went to Portland to visit Detlev Boysen, and I spent 7 hours with them, with Detlev and Ursula Sandow, who's a senior postdoc in his lab and really the one leading his experiments. Detlev has had an extensive history in studying particularly the role of adenosine, a neurotransmitter in the brain, and its potential anticonvulsant properties as a sort of naturally occurring anticonvulsant in the brain. And so his idea has been throughout his career that if you can find ways to increase the levels of adenosine, which are already in the brain, you're not introducing a foreign substance, that you could affect seizures by doing that without having to use drugs. And so we funded him a couple of times throughout his career as he's advanced the work on adenosine. And his last grant with us several years ago was on using some implants of silk that actually release adenosine in the brain. And he saw wonderful effects with that, but you also have this silk implant. So it was really a proof of concept study. We wouldn't be implanting silk into people. But in his current grant, he's taken that another step by looking at how adenosine modifies really the structure of our DNA. And by modifying the structure of the DNA, it changes the way genes are expressed. And his idea is that by modulating the way the genes are expressed, you're affecting the disease process. And he thinks that adenosine can play that role. So his most recent work is looking at different ways that adenosine modifies DNA, and if he can strategically change the way that adenosine is working to modify the DNA and therefore change the disease process. So again, it's sort of using the natural system that's going on within the cell, but changing it to our benefit to see if he can prevent the development of epilepsy after an injury.
So are there particular studies? I think some of his work I've read has been in injury.
Yeah, so he works in animal models of injury, but that is his area of interest. We know that brain injury is one of the causes of acquired epilepsy in humans. And one of our biggest problems in the clinic is that you don't know who is going to develop epilepsy until they start coming to the clinic with symptoms like seizures.
Okay.
So of all the people who are in car accidents or veterans who come back from war, it could be years before they develop symptoms of epilepsy, and we don't know who those people are. And so what he's trying to do is intervene with something that's fairly innocuous, and say, we know someone has had an injury. If we start treating them relatively quickly after that injury, can we decrease the chance that they will develop epilepsy? And that's what he's testing in an animal now.
It's amazing work. It really is. So what do you think the immediate future is for the doctor and his team? What's the next steps? Are you free to talk about that?
They are currently collecting data on their most recent studies, and I think that we hope to see some positive results from them that would tell us if they've been able to intervene in the disease process. before the animals develop seizures, if they can prevent a percentage of them from developing seizures, then I think they will have really hit on something that we haven't been able to do before.
Okay, now help me understand this too. Realistically, how long does it take before they're able, let's say they really are able to document some proven success in the laboratory animal studies? How long does it take before something like that is actually approved for human use?
It's a really good question. If this works, their next step, because they're using a drug that is not something they could use in humans right now, but if this works, they do have a plan to use some similar drugs that are already FDA approved. So that's helpful.
Okay.
Because repurposing a drug that already has FDA approval can fast track it to the clinic as opposed to going through the entire process from scratch. So, I mean, it still could be a couple of years away, but if you're looking at a molecule that is not FDA approved, you're looking at 5 to 10 years. Okay. So, I would hope that they would be able to fast track something like this to the clinic using an FDA approved drug if it's found successful in the animals.
One would certainly hope so, particularly with all the people coming back, especially in the military with the brain injuries. That would be just fantastic.
Absolutely.
If they could help those folks out as quickly as possible. I wanted to shift gears over to your one of your other visits was with Dr. Raimundo D'Ambrosio at the University of Washington. I know he has had some award-winning research in the epilepsy studies. What can you share about his latest work?
So Dr. D'Ambrosio also has a history with Cure in that, and he also has an interest in prevention of epilepsy after injury. He was a part of a team a number of years ago that demonstrated the efficacy of simply cooling the brain in the area of injury to prevent epilepsy. And what he's done from that is now looking at, what do you do when you cool something? And with tissue, you are inducing anti-inflammatory pathways. You're decreasing inflammation, and something about decreasing inflammation can halt the disease process. What he's done recently, which is really amazing, is that in trying to think about how would we do this in people effectively and easily and not having to do surgery repeatedly, is that he came up with the idea to take a patient who's going to have, usually a patient that's identified with a focal epilepsy, which means there is a particular part of the brain that the seizures are coming from. Those are people that usually would have that piece of the brain removed in surgery and hope that that can stop the seizures. He's working with a neurosurgical team to take the CAT scans from those patients, reconstruct them in a 3D modeling system, and then he does a 3D printed model of that patient's skull, so it looks exactly like the patient. And then using the information from the neurosurgeon would tell them which piece of the skull would be removed in order to do the surgery to remove the seizure focus from the brain. But instead of doing that whole surgery, his idea is that he would remove the piece of the skull over the seizure focus and replace it with a titanium aesthetic that is 3D printed, again, to match the exact curvature of that patient's skull. Because titanium as a metal naturally conducts heat. And so his idea is that if all you do is replace that piece of skull with a piece of titanium, you may effectively cool the brain enough to stop the seizures.
Amazing. Now, is titanium is the metal that the only metal that the human body adapts to, correct?
Exactly. He's using the same biocompatible titanium that's used in joint replacement. Mm-hmm. So we already know that it's safe to be implanted into the human body.
When I read your report, I found this absolutely fascinating. What are the next steps? Because this just sounds so logical. How does he take this to the next level, Julie?
So his next step is that they have a proof of principle study planned in human patients, in which they just want to ask the question, can replacing the skull cap with the prosthetic decrease the temperature at the surface of the brain? And so they are going to, in surgical patients who are already going to be having surgery and have a piece of their skull removed, they're creating these titanium caps. They're going to place them on the brain for 30 minutes during their surgical procedure and just measure the temperature at the surface of the brain. Then they'll take it off and the surgeon will proceed with their normal surgical procedure. But the idea is that if they can show that by placing the titanium on the brain, they're able to cool it by even, all they need is really 2 degrees Celsius, which is not very much. Mm-hmm. If they can show that that works, then they can apply to the FDA to really try this in patients long term and see if it affects their seizures. So and I think their plan is to start that proof of principle study to see if they can cool the brain with the titanium. In humans, I think their plan is to start in November, so very shortly.
And this is where I saw in your report they would apply for the humanitarian device exception.
Yes.
I think it's amazing though that the work he's doing, it's truly remarkable, and if that works, this could be a huge breakthrough, and I think this is where people really get excited. That now, would researchers like Dr. D'Ambrosio be able to get this type of funding support through normal channels, or is Cure's funding of a project like this exceptional?
There are a number of things that we have funded that others would not have funded. I think his initial work on cooling was not something he could have gotten funded elsewhere because it didn't really come from it probably wasn't seen as safe enough for a traditional funding body. We like to take risks. And additionally, the work he's doing now under our grant, which is not directly related but came from that, is that he is screening FDA-approved anti-inflammatory drugs in his model, and other funding bodies unfortunately aren't as interested in repurposing of drugs, even though for us there's great opportunity there in repurposing of drugs because they're already FDA approved for safety in humans. And but it's hard to get that work funded elsewhere. And so that also would not have been funded in other places. They went through a state-funded, in the state of Washington, biotechnology group to fund the patent and clinical trial on their 3D printing titanium project right now. So they were able to get some local interest for that. But we are proud to say that it came from his initial mechanistic work on cooling that we funded to show that it's possible to do this.
That's great, and I think your openness to fund that type of repurposing of drug research is just going to be so vital to the epilepsy community. Now, what did it mean for you to get out in the field, out in these labs, and see this research firsthand? How has it changed your perspective?
I loved doing this. I'm a scientist by training, and when typically our reporting out from our grantees is a written report twice a year, but it's focused on the aims of their grant that was written for us and providing us with an update. What we don't get is all the other qualitative information that comes with that. And by spending time in the lab with both the principal investigator but also their the people that are doing the work, and they have their hands on this stuff every day, I got a lot more information. I would not have gotten this story about his 3D printed titanium alloy from Dr. D'Ambrosio because we are not directly funding that project right now. So I wouldn't have heard about that in his progress report unless I went and sat with him in his office and he can pull out of his box and show me all of his stuff, which was so exciting. And getting to just really spend quality time. I spent multiple hours in each lab, anywhere from 2 to 3 hours up to 7 hours in each lab, and getting to see their space and their technologies and ask questions and meet the people. I learned so much about each of them and their work beyond what our dollars are directly funding right now. And that was really important for me.
That's amazing. And I know part of Cure's role is to really connect the dots between all these different research studies being done throughout the world. And do you see more possibilities as you're able to compile these reports to connect researchers to accelerate the progress?
Oh, we would love to do that. And that is something that I think as we grow, we will have more opportunity and more capability to do that kind of thing. But that's absolutely one of our goals, which is to, when we see synergies, is to help those people connect. And because we all believe that the next greatest breakthrough is unlikely to come from one single laboratory, that it's really going to come from people breaking down their walls and working together. And so when we see things that could complement each other, we do our best to try and bring those people together. We don't have a systemized way of doing that right now with our independent grant programs. Outside of we have some team programs, but we would love to be able to do that more. And I think that these kinds of visits really help with that, because you can see the whole breadth of an investigator's work, not just specifically what we're funding at that moment.
I can hear the excitement in your voice. It sounds like there's going to be some more travel in your future.
I can't wait to do this again. It was wonderful. It really was.
That's great. Thanks again for your time. I sure appreciate it, Julie.
Thanks so much, Jack. Nice talking to you.
Thank you for listening to Hope, Innovation, Discovery. We hope you'll share this episode with family, friends, and colleagues that may benefit from the content. For more information on Cure's research and the unrelenting commitment to curing epilepsy, visit cureepilepsy.org.